Welcome to my presentation. In this presentation, I'll be giving you a brief overview of my UROS project. The Undergraduate Research Opportunity Scheme has allowed me, a third-year medical student at Lincoln Medical School, to work with the Haematology Department of United Lincolnshire Hospitals NHS Trust, and also with the University of Lincoln's Joseph Banks Laboratory to carry out research over the summer of 2021. I was fortunate to have Professor Ciro Rinaldi and Dr. Matthew Simmons supervise me for this project. Although we had initially planned to carry out this research project over the summer of 2020, we had to postpone the project due to the COVID-19 pandemic. In this project, I looked at GATA1 expression in myeloproliferative neoplasms. MPNs are types of blood cancers which arise because of abnormal changes, known as mutations in the DNA of bone marrow cells, which ultimately produce blood cells. MPNs are a very rare form of cancer, and so we have very limited knowledge about the scientific basis of this disease in comparison to other common diseases such as diabetes. GATA1 is the name of the gene that I studied in this project. When I first submitted my UROS application back in 2019, we had made plans to study the difference in GATA1 expression in essential thrombocythemia, ET, and primary myelofibrosis, MF, two different subcategories of MPN. This was due to the reason that a previous study conducted at the University of Lincoln indicated that GATA1 expression could possibly be used as a diagnostic biomarker in the differential diagnosis of ET and MF. Simultaneously, we were planning to collaborate with research scientists at Robert H. Lurie Comprehensive Cancer Center at Northwestern University Chicago. They were planning to work on immunohistochemical staining on bone marrow trephines obtained from pre-MFs to patients and controls studied in my project. This project was planned to facilitate the exciting new collaboration between the University of Lincoln and the Division of Hematology at Northwestern University Chicago. However, these exciting plans that we had carefully premeditated could not go ahead due to the COVID-19 pandemic. As a result, I had to make slight alterations to the plans that I had initially made. Through my revised project aims, I wanted to evaluate whether GATA1 can be used as a diagnostic biomarker in the differential diagnosis of MPN. So what exactly is GATA1? GATA1 is a gene that plays a key role in the development of red blood cells and platelets. This gene mediates the maturation of red blood cells from their precursors, known as erythroblasts, and also is involved in the key stages that lead to the development of blood platelets from their precursors, including megakaryoblast, promegakaryocytes, and megakaryocytes. In order to investigate alterations in GATA1 expression in patients with ET, PV, and MF, peripheral blood was collected from patients with these subcategories of MPN. In addition, blood was collected from healthy controls to allow comparison. Thereafter, the FICOLL PAQUE separation technique was used to separate mononucleated cells from the peripheral blood that was initially collected. So what are mononuclear cells? Mononuclear cells are blood cells that have a single round nucleus, such as lymphocytes and monocytes. When isolated from circulating blood, they are called peripheral blood mononuclear cells, PBMCs, but other sources exist, such as umbilical cord, spleen, and bone marrow. The next step involved the extraction of RNA from the mononucleated cells that were separated from the peripheral blood in the last step. Trisochloroform extraction technique was employed to extract RNA. Following the extraction of RNA, copy DNA had to be synthesized. This step involved the usage of high-capacity copy DNA reverse transcription kit. All these steps allowed me to set up and run a quantitative PCR assay in the lab. Having completed the qPCR assay, I was able to input the results from the assay onto an Excel spreadsheet. This Excel spreadsheet allowed me to carry out important calculations and ultimately draw the two graphs that were displayed on my poster. As seen on the first graph on my poster, there was a clear one-fold decrease in GATA1 expression in MPN in comparison to the control samples. The second graph on my poster compares GATA1 expression in the three main subcategories of MPN studied in this project. As seen on the graph, GATA1 expression in PV was significantly higher compared to ET and MF. Furthermore, it is very important to note that expression of GATA1 was similar in both ET and MF. These findings have allowed me to conclude that GATA1 expression could potentially be used as a diagnostic biomarker in the differential diagnosis of MPN. This is because GATA1 expression seems to be altered in all three subcategories studied in my project. Since GATA1 expression is similar in ET and MF, a larger cohort size could improve the reliability of the data. This could also allow for t-tests or ANOVA tests. Overall, this project has allowed me to meet my project aims, and I can confidently say that my work has laid a foundation that can be built upon with more research and time. Although during the course of the project I had to face many challenges, including having to self-isolate and difficulties in obtaining fresh blood samples, this research opportunity has allowed me to gain invaluable laboratory experience, whilst allowing me to experience the realities of working in research. Not everything will work out as planned.